It's my pleasure to welcome you to the latest webinar in our Centre for One Health Spotlight series. My name is Terry Smith and I direct the Molecular Diagnostics Research Group here at NUI Galway, where our research focuses on the development and application of in vitro amplification technologies for the rapid detection and diagnosis of infectious disease. Our recent focus has been on the application of molecular diagnostic tests at the point of care. So the One Health concept recognizes that human health is tightly linked to the health of animals and the environment that we share. And the Centre for One Health here at NUI Galway brings together researchers and key stakeholders in human health, as well as animal health and environmental health, to tackle the leading health challenges that we face in today's world. So today's Spotlight Series webinar will focus on antimicrobial resistance, which is a global health challenge. And we'll hear today how this challenge is being addressed from a One Health perspective. Antimicrobial resistance is one of the most serious social and economic challenges and threats facing the world today. It's an increasing threat not only to human health, but also to animal health and the environment, having arisen mainly due to inappropriate or excessive use of antimicrobial agents. A frightening statistic, it's estimated by the UN Interagency Coordination Group on Antimicrobial Resistance that by 2050, unless action is taken, as many as 10 million deaths per annum will be attributable to antimicrobial resistance. So that's a figure that really would make you think. A number of zoonotic bacterial species, including among others, E. coli, Campylobacter and Salmonella, are commonly associated with antimicrobial resistance. And from a One Health perspective, the early detection of these organisms and important antimicrobial resistance markers in various settings of human, animal, and environmental relevance is critical for improved control and control of resistance, and also to reduce or minimize the spread. So over the next 90 minutes or so, we'll hear how One Health approaches are being investigated, aimed at improving the detection and control of the spread of antimicrobial resistance in humans, animals, and the environment. So today's webinar will involve 4 short presentations by Dr. Rohn Higgins, Dr. Alex Dimenezes, Professor Diane Morris, and Dr. Liam Burke. And their talks will be followed by a panel discussion and a Q&A session with the speakers. So if anyone has any questions that you wish to put to any of the panelists, please use the chat function, or if you prefer to use the Q&A function that Zoom has. So before I give you or introduce the first speaker, I just want to briefly introduce a European initiative that we're involved in, and that 2 of the projects will be focused on work related to this. And this is the European Joint One Health European Joint Project. Apologies. This project involves a consortium of 38 organizations across 19 European countries, with funding from Horizon 2020. The total budget for the program that's associated with this One Health European Joint Project is 90 million, of which 50% is being provided by the European Union and 50% is being provided by partners. It's a 5-year project, and its main objectives are to develop and support the European network of public mission organizations with reference to laboratory functions on infectious diseases. It involves institutions with a mandate from their national authorities or regional authorities, their ministries and or food agencies. And the partnership and the consortium aims to undertake cross-border public health, animal health, and food science collaboration, all focused on a One Health perspective and One Health solutions. Another objective is to improve prevention, detection, and response in the areas of foodborne antimicrobial resistance and emerging infectious disease threats. And then as well as integration and alignment of approaches across Europe through deciding on a joint research agenda. So it's an opportunity to facilitate the quality and compatibility of information for decision making across all sectors in the food chain. And this consortium and the funding it's received is supporting a large number of projects as well as training and CPD initiatives. And one project I want to briefly introduce, which I'm associated with as lead researcher, is called the WorldCom project. And that project is focused on the development of new tools for real-time detection of zoonotic bacteria and antimicrobial resistance in veterinary, human, and environmental sources. That project, which involves 6 partners, aims to develop new molecular diagnostic tools for early real-time detection of resistant pathogens in animals, as well as specific antimicrobial resistance markers, as well as looking in various environmental sources. Second objective is to develop diagnostic tools for zoonotic bacteria and antimicrobial resistance marker detection to be used on site, so to be used at the point of care. And linked to that is an aim to support the use of IT solutions that can be used as part of tools for the diagnosis of resistance infections in humans, animals, and various environments, again, at the point of care. So without further ado, I'll start the presentations, and I'll introduce our first speaker, Dr. Owen Higgins. Owen is a postdoctoral researcher in the Molecular Diagnostics Research Group, and he's currently working on the WorldCom project that I've just introduced. Owen has a lot of experience in the development of novel isothermal amplification technologies for infectious disease, as well as designing and developing infectious disease molecular diagnostics tests. He's the inventor of 2 novel isothermal amplification technologies that have arisen from his PhD research. And in today's talk, he's going to outline his ongoing work, which is focused on developing isothermal amplification-based assays based on a modified LAMP technology. You may have heard of LAMP technology, which is in the media at the moment. It's being used at Dublin Airport for rapid COVID-19 testing, and it's seen as a very rapid and useful tool for within hours detection and result versus the standard PCR. So Owen's using his modified LAMP technology for the detection of key antimicrobial resistance markers. So over to you, Owen.
Hello everyone, my name is Owen Higgins. I'm a postdoctoral researcher working with the Molecular Diagnostics Research Group at NUI Galway. And my talk today will be to give you an overview of our involvement in the WorldCom One Health EJP project. So our main objective in this project is to develop on-site tests for the detection of pathogens associated with antimicrobial resistance. We hope to deploy these tests in various agricultural and environmental settings such as farms and associated water systems. The sample types we'll be focusing on will be that of bovine, pig and chicken fecal matter, as well as irrigation water and drinking water. The pathogens that we'll be focusing on will be enterobacteria such as E. coli. So these bacteria contain genes which confer antimicrobial resistance, or AMR. So the AMR gene targets that we'll be focusing on will be extended spectrum beta-lactamase genes, or ESBL genes. and these are associated with resistance to the commonly used beta-lactam antibiotics. So to achieve all this, we essentially have two main research areas to focus on, and that will be to develop rapid on-site sample processing methods for the extraction of bacterial DNA, and also to develop on-site molecular diagnostic tests to analyze this extracted DNA. So for our sample processing method, we currently have an extraction protocol that we can perform in roughly 15 to 20 minutes. So this begins with your sample that you'll be testing. So for our animal fecal sample, we'll be taking swab samples, and for our water sample, we'll be using water filtration. So either your swab or your filter is transferred to cell lysis reagents, and this then is transferred to a heat source. So the combination of this will allow you to extract your bacterial DNA from your sample, and then an aliquot of this can be transferred to lyophilized reagents for on-site molecular testing. Now, as for our molecular tests, we'll be using DNA amplification methods. So these are essentially techniques that mimic DNA replication and allow you to amplify your gene target to a detectable level. This detection is typically done through fluorescence detection using DNA probes that are labeled with fluorescent labels, and then these reactions are monitored using fluorometers. So PCR is your gold standard molecular diagnostic test and has been the test of choice during the COVID pandemic. This method requires thermocycling, so it requires rapid reaction temperature changes from high heat to low heat, and it does this using thermocycling fluorometers. So for example, on the top right there is a Roche PCR machine. Now this is quite expensive and also quite large, so for on-site use it's quite impractical. So as an alternative to PCR, there are single temperature DNA amplification methods, and most popular method here is LAMP, or loop mediated isothermal amplification. Now LAMP has also been used as a diagnostic method during the COVID pandemic, and this is more for on-site testing. Now, because it's a single temperature method, it can be used with smaller, more portable thermostatic fluorometers. So on the bottom right there, you can see an example of such, the QIAGEN instrument. So this allows you to perform a reaction at a single temperature and then also record your fluorescence in various different fluorescence detection channels. So at Anyway Galway, we have developed a novel LAMP method called LEC-LAMP. This allows for real-time multiplex detection. So in a single reaction, you can detect multiple different targets, and it also allows for single base specificity. So if you had two gene targets that were only differentiated by a single DNA base, this method would allow for detection and differentiation between these two targets. So as for the AMR targets that we were going to target with our LEC-LAMP technology, we, as I said previously, we'll be focusing on ESBL genes. In particular, we'll be focusing on the CTXM group 1 genes, which confer resistance to cefotaxime antibiotics and are commonly found in pathogens such as E. coli and Klebsiella pneumonia. Now, there are two subtypes in this group. The first, CTXM1, is associated with animal infection, and CTXM15 is associated with human infection. So we need to be able to differentiate between these two. But unfortunately, the gene sequence for these two targets are almost identical and only differentiated by a small number of isolated DNA bases. But this is where our LEC-LAMP technology is useful. So we have developed a CTXM115 LEC-LAMP assay. So this is a duplex assay, which is essentially a single test that can detect and differentiate between CTXM1 and CTXM15. So far, we've evaluated this test using E. coli isolates extracted from environmental samples, and the results we have gotten have demonstrated fast, specific, and low levels of detection. So what you can see here is a fluorescent readout from one of our LEC-LAMP assays. We tested the assay with E. coli CTXM1 and CTXM15 at low level concentrations using 10 cell equivalents. And we recorded the reaction in two separate detection channels, the FAM detection channel and the HEX detection channel. These are essentially just two different color fluorescence detection channels. And what we can see is that we have differentiation between the two targets. So essentially your CTXM1 target is only detected in its appropriate FAM detection channel, and the CTXM15 target is only detected in its appropriate hex detection channel. And this just demonstrates our assay specificity. And also the fact that we are detecting down as far as 10 cell equivalents, it's indicating low limits of detection. And also these low limits of detection are detected within a 15 to 20 minute time period, which is ideal for on-site use. So as for testing our extraction method and the Lec lamp method on actual samples, we currently don't have fecal samples that contain CTXM1 or 15. So for the moment, we've been using spiking experiments. So essentially, we'll just take our bovine fecal material and spike this with a serial dilution of E. coli CTXM1, and then test this with our extraction method and our Lec lamp assay. And as you can see, we're able to detect down, so far, we're able to detect down as far as 100 cell equivalents and in around that 25-minute mark. So we still hope to optimize our extraction method and the Lec lamp assay to bring this limited detection down closer to the 10 cell equivalents, but we did expect some inhibition from the bovine fecal matter. But as a starting point, we're quite pleased with this. So the next step will be to test actual samples. So we'll be collaborating with one of our WorldCom partners, the Friedrich Loeffler Institute in Germany, and they'll be providing us with pig fecal samples containing CTXM1. And after this, then we hope to do some on-site testing. So once we have validated our extraction method and our Lec lamp assay, we then hope to send this out to some of our WorldCom partners. For example, the University of Surrey and the University of Madrid both have links with veterinary schools, so they will be able to further validate both of these methods. So before I finish, I'd just like to acknowledge the contributions to this work by Alexandra Schwery, one of our researchers, and also Dr. Louise O'Connor and Professor Terry Smith. So thank you very much.
Thanks very much, Owen, and well done in terms of keeping to time. So without any delay, I will move on to the next speaker and introduce Dr. Alex Dimenezes. Alex joined NUI Galway as a lecturer in soil microbiology in January 2017. He obtained his PhD at the University of Liverpool and then worked as a postdoctoral fellow at University College Dublin, and subsequently moved to the other side of the world to work at CSIRO in Australia before coming back to the UK and working, doing a research fellowship in the University of Salford. He's research focused on next generation sequencing, and he's published several studies using next gen sequencing to characterize microbial communities using 16S ribosomal RNA, gene sequencing, metagenomics, and metatranscriptomics. And his research projects, since he started at NUI Galway, he's developed a number of research projects focusing on a variety of areas of soil microbiology and looking at plant and animal microbiomes as well, including investigating relationships between radiation and soil microbial ecology at the Chernobyl exclusion zone. And I recall a conversation, I think he's traveled over there. Obviously, that's been disturbed by the whole COVID lockdown and travel prevention, but very interesting project. He's also a partner in another one of the One Health European joint projects, where he's investigating antimicrobial resistance gene transmission in wild animals. And Alex is going to outline the aims of the FED Antimicrobial Resistance Project and his role, including his sequencing approach using antimicrobial resistance gene enrichment probes. So Alex, over to you.
Hello everyone, and thank you for coming to listen to my presentation. My name is Alex Dimenezes, and I'm a lecturer in soil microbiology here in NUI Galway. So I will give you a talk about the project I'm working with, which is entitled FED-AMR, which is also part of the One Health European Joint Programme on antimicrobial resistance. So my project is particularly focused on working with or studying the role of free extracellular DNA in the dissemination of antimicrobial resistance over ecosystem boundaries along the food and feed chain. So that's the long title for my project. And this project is part of the second call for research projects of the One Health European Joint Programme. It's a large project with 13 laboratories and 12 partner institutions across Europe. The PI is Werner Ruptisch in Austria, in AGES, which is a research institute that belongs to the Austrian government. Our project is particularly focused on, like I said, looking at extracellular DNA and the controlled conditions, and particularly looking at model farms in Austria and also in Portugal. So what we're interested in is in looking for the antimicrobial resistant gene concentrations, diversity, dynamics, and variability in an annual longitudinal study covering crop growing periods, different fertilizations and land management techniques, and various but interlinked environmental compartments. So at NEOIG, I'm one of the main people involved in the project, but I also have recruited Caritini to help me with this project. Dr. Gavin Collins is also a part of the project, as is Dr. Florence Abram and Dr. Colin Lawton. So the background to the project is that most research projects studying antimicrobial resistance in the environment are focused on the role of live bacteria in the spread of antimicrobial resistance genes. However, antimicrobial resistance genes may also be present in the environment in the form of extracellular DNA. So this is DNA that's not contained within bacterial cells or any cells. So antimicrobial resistance genes can be encoded in the extracellular DNA, and because the extracellular DNA is not in a cell, you don't require cell-to-cell contact for the transmission of antimicrobial resistance genes. So extracellular DNA can serve as a high-risk source of resistance determinants in agricultural soils and along the food and feed chain. So here we have a diagram showing normally what you have is a situation where the bacterial cell has the antimicrobial resistance gene in its genome, and usually that, for horizontal gene transfer to occur, you would have two cells coming close together, and then you have the transmission of antimicrobial resistance genes to the donor cell. But in the case of free extracellular DNA, you don't need to have this close contact between the bacterial cell and the recipient cell. So our hypothesis is that horizontal gene transfer of antimicrobial resistance gene through the free extracellular DNA overcomes ecosystem barriers and bottlenecks, meaning that the AMR genes can cross from different farm compartments or different environmental compartments or habitats, such as from soil to the gut ecosystem in farm animals and also humans. And also we believe that high microbial diversity may constitute a barrier for the spread of AMR genes. So our aim is to evaluate the role of natural bacterial transformation relative to bacterial conjugation in the dissemination of AMR over environmental ecosystem barriers. So we have several objectives, and here in NUIG, we're particularly interested in the objective 1, so determination of the rhizostome and microbial biodiversity in 11 environmental compartments, and identification of the role of extracellular DNA for horizontal gene transfer in the tested compartments. There are other objectives which are part of the FED-AMR project, which are going to be the topics focused by other members of the project, but we're particularly interested in objective 1, and objective 3 as well, so the identification of selection pressures in these 11 compartments, and we're also going to be working with objective 4, so the identification of environmental conditions modulating transformation frequencies in porcine chemostat gut models. So we are currently performing sequencing, we're collecting samples to sequence the antimicrobial resistance genes in various samples taken from many countries, particularly in Austria and Portugal, but also here in Ireland. We're going to determine the microbial diversity and the presence of antimicrobial resistance genes. And we're also going to be collaborating with our partners, which are also looking at the role of Clostridium difficile in antimicrobial resistance gene transmission. So this plot here shows the compartments that we're interested in. So our focus is on pig farming, and we have access to 2 model farms, one in Austria and one in Portugal, where we're going to look at the antimicrobial resistance genes in the extracellular DNA in all of these 11 compartments here. So the manure, on the soil, on wild animals, on the pig in the farms, on the feed that is given to the pigs, on the crops that are growing in close proximity to pig farming, on the farmers themselves, on the water that's in the fields, surface water, irrigation water, also on wastewater treatment plants and drinking water. So we're going to look for antimicrobial resistance genes in all these compartments, particularly focused on extracellular DNA. Here is just a map showing the open-air laboratory in Austria, where we have plantations of several different crops. So we have maize, we have mainly maize, but also winter wheat and other crops that are grown. And we have pig farming not far from here as well. So we are looking to see whether antimicrobial resistance genes are crossing between these different fields and the different ecological compartments through extracellular DNA. Here at NUIG, we are particularly interested in collecting wastewater samples from Mutton Island Wastewater Treatment Plant. We are collecting wild deer feces from Potumna Forest Park. We are going to perform DNA extractions and then send those DNA for sequencing, particularly focused on environmental DNA, but also total DNA. And we are particularly in charge of performing the bioinformatic analysis for the over 400 metagenome samples that are going to be generated from this project. So in more detail, what I mean by our sequencing approach, what we are really interested in is in a novel strategy to quantify the diversity of antimicrobial resistance genes. That is because what we tend to do for characterizing antimicrobial resistance genes in the environment relies on basically taking all the DNA that we can extract from a particular environment, so a particular environmental sample, let's say soil sample. We take the soil sample and we extract all the DNA that's present in the soil. And then we sequence all of that DNA and we use bioinformatic techniques to identify the antimicrobial resistance genes that are present in these samples. However, there's a problem, which is that even samples that are very contaminated with antimicrobial resistance genes, those antimicrobial resistance genes are still hard to find using these techniques because the majority of the genes that are present in these samples are not antimicrobial resistance genes and they're not the target of our study. So in recent years, we've had some developments in this field. So there have been a couple of papers, a couple of studies which have demonstrated that it is possible to enrich our samples for antimicrobial resistance genes using probes which target the antimicrobial resistance genes and allow us to increase our coverage of these genes so we can quantify a greater number of antimicrobial resistance genes. This is a new strategy that was developed recently by 2 research groups, one in Spain and one in Canada. And when I joined the FED-AMR consortium, when we were writing the proposal, I particularly mentioned this approach, and I said that I believe that this would be a good way to go for the FED-AMR project, because that would give us a much better coverage of the diversity of antimicrobial resistance genes in the environment. So this approach uses magnetic beads, which are very small spheres which have magnets in them. And these beads have DNA, little pieces of DNA that can basically grab environmental DNA of interest, in this case AMR genes, and then we can use magnets to pull those beads out of our samples, and these beads will then contain an enriched amount of the genes of interest, in this case antimicrobial resistance genes. This means that when we perform our sequencing, we will have a much greater number of AMR genes to investigate, and we can quantify and determine the dynamics of these genes in the different compartments to a greater detail than has been previously the case. So I want to thank you for your attention. You have there my email address, so you can contact me if you have any further questions. So thank you, and bye-bye.
Thank you very much, Alex. Appreciate that. So I will now move straight on to our third speaker, Professor Diane Morris. So Diane is a colleague and a research collaborator on the WorldCom project. She's Professor of Antimicrobial Resistance and One Health at NUI Galway and head of the discipline of bacteriology in the School of Medicine here. She established the Antimicrobial Resistance and Microbial Ecology Group back in 2010. And in addition to directing that research group, she's also the current director of the Centre for One Health here at NUI Galway. Her research group works closely with national and international research groups and other agencies, focusing on antimicrobial resistance, on food and waterborne pathogens, emerging contaminants, and also on the wider societal impact of infection and One Health. Diane is leading the 4-year jointly funded EPA-HSE ARREST project, which stands for Antimicrobial Resistance and the Environment, Sources, Persistence, Transmission and Risk Management. She's also the lead researcher on an EPA-funded PEER project, which stands for Public Health Impact of Exposure to Antibiotic Resistance in Recreational Waters, which we'll hear about in the fourth presentation from Liam Burke. So Diane will outline the work being undertaken by a research group on the ARREST project, generating national level data on the key sources, hotspots, and the drivers of antimicrobial resistance in the environment from various sectors. So over to you, Diane.
Good afternoon, and thank you to the organisers for giving me the opportunity to present on the ARREST project. This project is a 4-year project funded by the EPA and HSE. Antimicrobial resistance is very much a One Health challenge. As you know from earlier talks this afternoon, the One Health approach recognises that the health of humans, the health of animals, and the health of the environment are interlinked. It is recognised globally that antimicrobial resistance is one of the greatest human health challenges we face in today's world. And it reached the floor of the UN General Assembly in 2016, only the 4th time in history that a human health issue has been brought to that forum. Following publication of the World Health Organization's Global Action Plan on Antimicrobial Resistance, governments around the world published national action plans on antimicrobial resistance, including the publication of Ireland's National Action Plan on Antimicrobial Resistance in October 2017. It is increasingly recognised in these action plans that there is a need to better understand the role of the environment in the transmission and persistence of antimicrobial resistance. As you may know, antibiotic resistance happens because we use a lot of antimicrobials both in human and veterinary medicine, and sometimes not always correctly. It was great to see the publication of Ireland's first One Health approach on antimicrobial use and antimicrobial resistance in January 2019, which brought together data on antimicrobial use and antimicrobial resistance in humans and animals. And it is good to see that further revisions of this report will include environmental data, including data generated by the ARREST project. As we know from this data, we use a lot of antibiotics in humans and animals in Ireland. Most recent data reveals that Ireland is ranked 9th highest in terms of our antimicrobial use in humans and 17th highest in terms of our antimicrobial use in animals. So when a human or an animal is given an antimicrobial, it is not always metabolized by the body. Some of that antimicrobial can enter the environment. If there's antimicrobial resistant bacteria present in the gut of that human or animal, there's also potential for those organisms to enter the environment in the waste. Generally, human waste is treated before being emitted to the general environment. But as we know from the most recent EPA urban wastewater report, there's still 36 locations around Ireland where raw sewage is emitted into recreational waters. We know that land spreading of waste is an important part of the circular economy, and we know that generally animal waste is spread on agricultural land in Ireland and elsewhere in a largely untreated state. But if there is antimicrobials and antimicrobial resistant bacteria present in this waste, there is potential for it to enter our groundwater streams, enter our surface waters, which if they act as sources of our drinking water supplies, or if they are used for recreation or for irrigation of food, there is potential for transmission to humans. So we know that antimicrobial resistance an examination of the environment and its role in the persistence and transmission of antimicrobial resistance is important. We know that the emission of raw sewage into recreational waters is not a good idea. And we proved this in 2017 through the publication for the first time of the detection of CPE in seawaters in Europe. We know the current wastewater treatment processes based on previous EPA-funded research does not actually remove all antimicrobial resistant organisms. And again, we also know that land spreading of biosolids and agricultural waste is an important part of the circular economy. We better need to understand what waste treatments are effective at removing resistant organisms and indeed antimicrobials from waste. So the ARREST project is very much taking a One Health approach. It's bringing together experts in human health, animal health, and the environment to examine the role of the environment in the transmission and persistence of antimicrobial resistance. So what we're actually doing in the ARREST project is we are going to map the drivers of antimicrobial resistance in the environment in 4 local authority areas: Galway City Council, Galway County Council, Cork County Council and Fingal County Council. We're going to assess the relative contributions of healthcare and agriculture to antimicrobial resistant organisms in the environment. We're going to assess the efficiencies of treatment processes for the removal of resistant organisms from drinking water, wastewater and manures, and develop a risk ranking protocol to assess the relative contribution of the various sectors on the sources and levels of resistant organisms in the environment. So the first step in this was to actually examine the role of the natural aquatic environment in the dissemination of antimicrobial resistance. So we carried out a scoping review which was published in Water Research earlier this year. We then wanted to carry out a mapping process to guide our sample location. So again, we're focusing particularly on Galway City and Galway County Council areas, Fingal County Council and Cork County Council. So if we take Cork County Council as an example, what we started with was mapping the potential sources of resistant organisms. So we mapped the locations of hospitals, of long-term care facilities, of areas of intensive agriculture, and we could then overlay this with the water network, essentially the potential transmission routes. And the mapping process is so powerful that it would allow us to drill down essentially to GPS locations. So that could then guide our optimal sample locations. And this mapping exercise was published in last year in the journal Antibiotics. So what we did then in the point prevalence survey and what we were carrying out in the longitudinal survey is we collected samples of sewage from hospitals, from nursing homes, from airports. We collected samples of influent and effluent from wastewater treatment plants and from integrated constructed wetlands. We collected raw waters from drinking water treatment plants and raw and treated waters from group water schemes. And we collected samples from estuaries, rivers, lakes and seawaters. In total, as part of the point prevalence survey, so this is a broad sampling survey that we carried out over a 12-month period. We collected 70 samples and we assessed these for the presence of resistant organisms and found ESBL producing Enterobacteriaceae, a particular type of antimicrobial resistant organism in 39 of the 70 samples, and CPE, a very concerning type of antimicrobial resistant organism, in 9 of those samples. It's interesting to note in terms of the influent and effluent from the wastewater treatment plants and the integrated constructed wetlands that resistant organisms were just detected largely in the influent. So we're now in the process of carrying out a longitudinal survey and further characterization of these isolates is ongoing. It's interesting to note if we look at the sewage and water isolates collected that there is a significant amount of multi-drug resistance, so resistance to more than one antimicrobial agent in both sets of isolates collected from both sources, including resistances to antibiotics of human health importance. So in task 3, what we're doing is looking at the role of land spreading in the dissemination of antimicrobial resistance into grasslands and soils. So we're collecting DNA from the manure that's applied to the grasslands, collecting samples from the grass and also from the soil, and carrying out analysis to compare them. We also assess the ability of integrated constructed wetlands to remove resistant organisms from farm effluents. So we collected 96 samples over a 1-year period, 48 influence samples and 48 effluent samples from 4 farms. And from these, we collected 86 ESBL producing isolates, again, a particular type of resistant organisms. But again, there was a significant difference between the influent and effluent. So this suggests that integrated constructed wetlands may be useful in the removal of resistant organisms from wastes. We also assessed the impact of anaerobic digestion on the removal of antimicrobial resistant organisms from manures. And again, we found that based on the total solids present in that manure, on average, when we spiked a sample of manure with a particular type of resistant organism, on average it was removed after about 7 days. And this work on anaerobic digestion was published earlier this year. So we're also developing a risk ranking protocol to assess the relative contribution of the various sectors on the sources and levels of resistant organisms in the environment. And we are in the process of developing a health model and an agricultural model. So overall, the ARREST project will generate national level data on the key sources, hotspots and drivers of antimicrobial resistance in the environment from various sectors, which will inform priority areas for action. It will provide evidence on the extent of contamination of the environment with resistant organisms to support policy decisions and help better engage with agriculture, local authority sectors, and indeed health sectors and AMR. We plan to re-input the data generated onto the maps, so this will provide engaging visual representations of our data to better enhance our communication with the public and policymakers. And overall, this will embed the One Health concept and build the capacity of Ireland's research community to support Ireland's National Action Plan on Antimicrobial Resistance. So we've done a lot, but there's still a lot to do. There is a need to develop systematic surveillance of resistant organisms and antimicrobial residues in the environment. We need to better understand what impact the different levels of wastewater treatments have on the removal of resistant organisms and antimicrobial residues. We need to better adopt a One Health approach, much like we see in the One Health European Joint Programme, towards funding of research in this area. We need to develop better mechanisms of translating our research into public policy, and through activities such as the Centre for One Health Spotlight series, enhance our communication of the One Health concept and indeed the issue of antimicrobial resistance with the public. So finally, I'd just like to thank the funders, the EPA and the HSE, for funding the ARREST project, all of the members of my own research group, the ARM group at NUI Galway, and all of the project team members of the ARREST project. And I'd be happy to take any questions you may have in the chat.
Thanks very much, Dervla. We'll hold questions obviously until the discussion forum and the question and answer session. So I'll move straight on now to our fourth and final speaker this afternoon. Dr. Liam Burke, who's a lecturer in bacteriology in the discipline of bacteriology here in the School of Medicine at NUI Galway, and he's also a member of the Centre for One Health. He's a microbiologist with research interests in antimicrobial resistant and zoonotic bacterial pathogen, and his research focuses on understanding their epidemiology and developing novel interventions to protect public health, including diagnostics, therapeutics, and also engagement-based behavioral change interventions. He's a co-principal investigator on the EPA's funded peer research project, and he's also a collaborator on the One Health European Joint Project, WorldCom project, myself and Dervla and Louise O'Connor collaborating with my lab. So Liam is going to discuss progress in the PEER project, which is investigating the possibility of being colonized with superbugs, that scary word, from using our lakes, seas and rivers for recreation. And he'll also discuss, I suppose, the question of whether people who regularly swim, surf, or do other in-water activities in rivers, lakes and seas are more likely to pick up and carry antibiotic resistance bacteria than those who rarely or never get in the water. So I'll hand over to you now, Liam.
Hi, my name is Liam Burke, and I'm going to talk to you today about the PEER project, which is being led by NUI Galway, and it's looking into whether recreational water users are at risk of picking up superbugs. So as you've already seen by now, antimicrobial resistance is a big problem, and it is a One Health problem. So it's a problem for human health, animal health, and also environmental health. And all of these different niches have to be taken into account when dealing with the problem of antimicrobial resistance. And I suppose one of the ways which antimicrobial resistant bugs can end up in our environment is through the release of wastes such as slurry and sewage. So this can happen when slurry is spread on land, or it can happen when sewage is released into our seas or our rivers. And it can also happen inadvertently in some cases when we have heavy rainfall, which occurs, as we know, a lot in Ireland, which can wash this slurry into our waterways, or it can also cause storm drains to overflow where wastes which would normally be treated through the wastewater treatment plants would end up getting into our waterways instead. And the impact that this can have is significant. So we can see here that there are still 35 raw sewage discharge points around the country. And if we overlap the points where the sewage is being discharged with the points where the Irish bathing waters are being tested, we can see that in the areas that are less than excellent, they often overlap with places where raw sewage is being discharged. This may not be a coincidence, and it may also indicate that the method that we are using to test the quality of our bathing waters may be suboptimal, because most of these bathing water are still being deemed sufficient for bathing, and we're not really sure whether that is the case. So a project was carried out in the UK by collaborators of ours on the PEER project, Dr. Anne Leonard and Professor William Gaze, and they found that people who frequently use UK coastal waters to surf were at a higher risk of being colonized by drug-resistant bacteria, specifically E. coli that are resistant to third-generation cephalosporin antibiotics, which are some of the most commonly used antibiotics to treat serious E. coli infections of the bloodstream and urinary tract. So this, combined with our finding of antimicrobial resistant bugs in bathing waters around the country as part of the ARREST project and previous projects, led us to believe that there may be a public health impact associated with bathing in these waters. So the PEER project then aims to investigate what is this health and well-being impact of exposure to antimicrobial resistant organisms in our recreational waters. And our main objective really is to support the development of new monitoring strategies and policy for AMR prevention and control in the environment. So some of the questions that we want to answer in the PEER project are: what are the public health implications of exposure to these bugs in recreational waters? And do our current bathing water quality regulations sufficiently protect public health? We also want to identify whether recreational water users are more likely to be colonized or infected with antimicrobial resistant organisms, as was shown to be the case in the United Kingdom. And if they are infected, we want to investigate how long these bacteria actually persist within the guts of these water users, and whether there's a difference in the length of time that they persist in water users who frequently use the water versus people who rarely or never use the water. We also want to investigate what the barriers and enablers are to people using natural waters for recreation. We're not trying to discourage people from using natural waters for recreation. We think that it's one of the greatest things that you can do for your health and your well-being. We really want to find out what is stopping people from using these waters, and can we do anything about it to increase people's recreational use of our natural waters. So what specific antimicrobial resistant bugs are we looking for? Well, you've probably heard a bit already about CPE, carbapenemase producing Enterobacteriaceae. So they are the silent public health emergency that's still raging in Ireland since 2017. And you can see in the graph on the right there that the incidence of CPE in hospital inpatients has increased steadily year on year, despite the best effort of the National Public Health Emergency Team to stop it. The WHO, the World Health Organization that is, also lists these bacteria as their first priority critical pathogen for which new antimicrobials are sorely needed. As well as CPE, we're also interested in detecting ESBL producing Enterobacteriaceae. So these produce enzymes called beta-lactamases that break down beta-lactam antimicrobials, which are some of the most commonly used antimicrobials. And these can also cause very difficult to treat infections that sometimes need the last resort antibiotics to be used. And of course, these antibiotics, they have serious side effects in certain patients, and they really are not that suitable for day-to-day use. So what are we going to do in the PEER project? Well, there's two parts. The first part is the colonization study, and this involves myself, Dervla, and our PhD student, Maeve. And what we are looking to investigate is the rate of carriage of these antimicrobial resistant organisms in a large group of people who frequently use natural waters. And we want to compare that then to people who don't use natural waters or who very rarely use natural waters for recreation. So we're going to do this by taking stool samples and analyzing the stool to see whether there's antibiotic resistant bacteria present. Then the second part, I suppose, of the colonization study is to see if there are bugs present, how long do they persist? And we will do this by taking multiple samples in our group of participants over the course of 2 years. So we're going to ask participants to provide us with one sample every 3 months over the course of 2 years, and we'll be able to tell then how long these antibiotic resistant bugs persist in their gut flora. So these are the methods that we're going to use. So we've developed these kits that contain these little scoops here that people can use to take a stool sample after they use the toilet. And they can send that back to us in the lab along with a survey with some information on how frequently they use the water. And we can culture their stool on these agar plates which contain antibiotics so that only antibiotic resistant bacteria will grow. Then after we've identified the antibiotic resistant bacteria, we'll do susceptibility testing against other antibiotics to see which antibiotics they're resistant to. And then we'll use molecular methods to detect the different resistance genes present. And we will compare the genomes of the interesting organisms that we find to those found in the ARREST project. So we'll compare what organisms we find in water users and non-water users to what organisms we find in the environment. So in the second part of the study then, the systems modeling study, Dr. Sinead Duane is going to examine the complex dynamic web of interconnected environmental, health and socioeconomic forces that impact water quality in Ireland. And she's going to do this by doing a literature review and doing some key informant interviews with stakeholders from the water quality field. She's also going to disseminate an online survey for all water stakeholders in Ireland, and she'll use this to generate a systems map where priorities for improving water quality can be identified with a view to future interventions. So an update on how we're doing in the project. So we had a big public engagement campaign just around the time of the lockdown on social media, and it was very successful. We also had great coverage in the press in Ireland and some international papers as well. As you can see here, we were in most of the national papers and some local papers, and we were also on the radio as well. And all of that resulted in a huge recruitment for our project. So a massive number of people signed up for the project. Over 1,088 citizens signed up from all over the country. So at least one person from every county in the island of Ireland is represented, and many of the coastal counties have a high number of participants, as you can see from this map here. So we are currently in the process of completing recruitment by calling all of the people who registered, and we've currently registered over 500 water users and about 80 non-water users to the project. So we sorely need more non-water users to sign up for the project, but we're working on that. As you can see, we've got a lot of water users in many different categories, but for the most part, they are open water swimmers. So if you're not much of a swimmer or a recreational water user, but you're interested in a citizen science project that may help to protect the environment, then you are welcome to join us. So please visit our website, www.nygalway.ie/peer, and you can sign up as a non-water user for the peer project. You can also follow us on our social media accounts here to keep up with all the news on the project. So all that's left for me to do is to thank my collaborators on the project. So they're listed here from 4 organisations: NUI Galway, the Health Service Executive, the University of Exeter and Queen's University Belfast. And thank you very much to the Environmental Protection Agency for funding the peer project. And I look forward to speaking to you all. Thank you.
Thanks very much, Liam. And thanks very much to all 4 of our speakers for keeping to time so well. So we've got about 30 minutes, maybe even a little bit over 30 minutes, depending on the level of interest and questions that come in for our question and answer. So first of all, thanks to all the speakers, not only for keeping to time, but for very interesting and informative presentations. And they'll all stay and participate in the panel discussion and Q&A session. So I'd also like to thank those of you in the audience who've submitted questions through the chat function, and invite anyone to feel free to continue to put questions into the forum based on the talks that you've heard, or even based on the discussion. And we'll do our best to get through as many questions over the next half hour or so. So maybe I'll kick things off with a question for each of our panel members in turn, and I'll start with Owen. So Owen, firstly, so you talked about Enterobacteriaceae such as E. coli and associated ESBL genes. Are there other antimicrobial resistant pathogens and genes that we need to be very concerned about, and that you might target within the WorldCom project?
Sure, Terry. Our research group and the associated WorldCom partners have looked at other ESBL-associated antimicrobial gene targets that are of high clinical importance. And some that have been identified would be, there's 3 in particular. There's OXA48, KPC2, and NDM5. Now, in terms of these 3 targets, we don't need to differentiate these in the same way we do with CTXM1 and 15. They're not too similar, but we do hope to apply our LECLAMP technology to these to develop a multiplex LECLAMP assay. So in a single reaction, detect each one of these targets, and then roll this out in the same way we have with the CTXM LECLAMP assay. And we hope to do this sometime in the new year.
Okay, thanks very much for that. I'll move on to Alex. And Alex, you presented some interesting stuff on investigating environmental DNA and its potential role in spread of antimicrobial resistance. So, I mean, in terms of the FED-AMR project that you're working on, how will the data generated from that project help with mitigation of antimicrobial resistance in farming? And I guess also there's a linked connection to the environment, as we've been hearing from Dervla and Liam, in terms of effluent that may get into the water system.
Yes, thanks, Terry. So the rationale of the FED-AMR then is that the risk of extracellular DNA as a source for antimicrobial resistance genes is, I'm sorry, I'm just getting some noise in the background, but it's basically not taken into account when developing strategies to prevent the spread of AMR. So when government bodies or research bodies are giving guidelines to governments about, how should slurry, a pig slurry they're going to spread on a farm, how should that be treated before you spread it? You need to know, first of all, are there AMR genes in there? And what's the risk of these AMR genes spreading to the native microbial community or to the microbial community that may end up being taken up by the animals that are then grazing on these environments? So we've been, the tendency for us is to look at the quantification of AMR genes by various molecular methods or microbiological methods. And we can, use metagenomics to determine if those genes are present in the DNA. But typically, we don't look at the environmental DNA. We don't look at the extracellular, not environmental DNA, but the extracellular DNA that's present. So that means that we may be overlooking, a potential pool of genes that may be driving the spread of these AMR genes. And this may be particularly important. For example, if you sterilize, if you kill bacteria, if you use some steps to try and decrease the amount of bacteria in a sample or partially kill them, or try to eliminate the bacterial community from a sample, you may still have the genes present in the extracellular DNA pool. And what we want to know in the fed AMR is this pool of AMR genes still a problem for farming? And we don't know yet because we're starting off with the experimentation and sampling and sequencing, but this is what we want to find out. I mean, does the extracellular DNA represent a risk? And if so, how can we mitigate this? So the first step is to determine whether there is a risk. And so far, our initial results suggest that yes, we can find AMR genes in extracellular DNA in these pig farms that we're working on. And the next step then is to see how risky that is, how widespread, and also link the AMR genes that we find in the extracellular DNA in the farms to the AMR genes that we find then in the guts, the feces of the pigs, and in the all the compartments that we're measuring. But the key thing here is to determine whether the extracellular DNA is a risk for the AMR gene spread, because then that'll have to be integrated into policies for containment of AMR genes.
Okay, thanks very much, Alex. I'll come back to you because this question has come in from our audience, but I'll just quickly move on and ask Dervla. It's been recently highlighted in the media that raw sewage has been discharged into the environment, and it's sort of scary when you see it on the news and think about the possible impact of that. And there's 35 towns, I think, around the country where there's raw sewage discharge. What are the potential impacts of that and the implications from an antimicrobial resistance perspective, Dervla?
Thanks, Terry. Yeah, I mean, it is concerning that there's 35 locations still where there's raw sewage being emitted into the environment, but it is an improvement on previous years. So every year there is an improvement. There is another location that's taken off that list, which is great. And thanks to the hard work of Irish Water in that regard. But it does have big implications for AMR, as I mentioned in my presentation, and also as Liam referred to. We, through work by Bláthnaid Mahan, one of our previous PhD students, she reported for the first time the detection of CPE in seawaters in Europe. And CPE are a very concerning organism because they're an organism that some strains of which are resistant to pretty much all of the antibiotics we have available to treat infection. And when we did the study, or when Bláthnaid did her work, she found that it was related to the sewage being emitted into the water. That's where this CPE in the water was coming from, was from the raw sewage that was being dumped into the water at that location. And that's what led essentially to the ARREST project and subsequently to the PEER project. There's a need to better understand what the implications of having antimicrobial resistant organisms present in recreational waters are, but there's also a need to stop them getting in there in the first place. And it's not a good idea to having raw sewage being put into waters where people are swimming.
Okay, thanks very much, Dervla. And then moving on to Liam, and we've heard from both yourself and from Dervla about CPE resistance. Can you tell us about the other kinds of superbugs you're looking at? For example, are you looking at ESBLs? We've heard about ESBLs from Owen. So is that part of your work plan as well?
Yeah, so ESBLs are the other type of superbugs, if you will, that we're looking for. And even though they're probably not quite as dangerous as CPE, because there are still some antibiotics that will work against them, they are still resistant to a lot of different antibiotics that are commonly used. So this has implications for increased cost, I suppose, because the drugs are more costly, and as well as that, it can delay successful treatment. So when the first line antibiotic is given, it doesn't work, so they have to go on then to use another one next. And often the bacteria that have these ESBL genes can also pick up other antimicrobial resistance genes on the same mobile genetic elements as well. So they can have ESBL genes and carbapenemase genes at the same time, and sometimes other resistance genes for different antibiotics as well. So because they're so mobile, like Alex was talking about with these mobile genetic elements that can be present in the environment, they can really pass these bugs around. So wherever there's antibiotics present, there's a kind of a need, I suppose, for these bugs to have these antibiotic resistant genes. So if we can kind of better control how we were releasing our antibiotics, then probably we'll have less antibiotic resistance.
Okay, thanks very much, Liam. So I'll move on. There's some questions have come in from our audience. So firstly, for Owen, is LAMP sensitive enough to be able to pick up single base? I guess he means LEC LAMP, your modified LAMP. Is it sensitive enough to pick up single base mutations that confer resistance, for example, to ciprofloxacin due to GyrA mutations?
Sure. So our LEC LAMP technology is quite new. The publication went out this year, and our initial testing was on synthetic targets. So its involvement in the One Health project is the first time we've actually used it on actual samples and actually that have actual SNPs or point mutations. But from our research, our study on LEC LAMP, it doesn't inhibit the sensitivity of your standard LAMP reaction. So I don't see any reason why it shouldn't be able to be sensitive enough to pick up any point mutations associated with AMR.
Okay, thanks for that. Another question's come in. I think this is for Dervla. Is there a sense of what proportion of the time organisms in the environment acquire antimicrobial resistance genes via transformation? Could be for Alex too, maybe, via transformation versus conjugation, knowing that some organisms are more competent for DNA uptake than others. So I'll open that up maybe to everyone. So any idea of what proportion of the time organisms in the environment acquire genes via transformation versus conjugation?
It's an interesting question and something that is being studied by a number of groups. For transformation to happen, you need certain things in place. You need the actual gene to be extracellular, which is what Alex I'm sure will come in on this as well. But you also need the bacteria to be competent, and there's very few bacteria that are naturally competent. Now they can become competent in the presence of certain substances, including antibiotics. But generally, now there have been a number of interesting studies that have shown that, for example, PAR-C and GyrA genes and streptococci, which confer fluoroquinolone resistance, can be transmitted via transformation. But again, it's for certain species of bacteria would only be naturally competent, like streptococci, Neisseria, and Haemophilus. So it is interesting. It's not that common is my understanding for certain species of bacteria that we've spoken about a lot today, like the Enterobacteriaceae, such as E. coli and Klebsiella. But for other species of bacteria, it would be an important mechanism of transmission. How it compares to conjugation for those particular species, I don't have figures, but it would be more important for some species than others. And again, Alex may have further comment on that.
Yeah.
Do you want to follow on from what Dervla said, Alex?
Yeah. So So generally speaking, I think conjugation is considered more important and it's more studied as well than transformation as a route for transmission of AMR genes in the environment. So I suppose that at least with fed AMR, we're not, I think this study is not there to kind of suggest that transformation is more important than conjugation. It's mainly to quantify the risk that transformation may be a problem. So that's why we are particularly targeting extracellular DNA, because since a lot of the studies so far have been performed either by looking at the spread of AMR bugs or horizontal gene transfer through conjugation and potentially transduction, but not so much through transformation. So the fed AMR is kind of trying to do exactly that. It's trying to find out how important this route for AMR spread may be. The part of the reason for our focus is because if transformation is a route under certain conditions for AMR spread, then policies for mitigating that need to take that into account. Because, like Liam was saying, that other factors may drive the spread of these AMR genes, such as the presence of antimicrobials in the environment that would create a selective pressure for the spread of these genes. But the dynamics that will drive the AMR spread AMR gene spread between live bacteria may be different for conjugation versus transduction versus transformation. So I guess what we're trying to do is determine what's the risk of transformation as a risk for AMR gene spread. And we are quantifying the AMR genes in extracellular DNA in various compartments. And there's also one of the groups involved in the fed AMR project in the UK, in Surrey, University of Surrey. They are going to look at transformation in model organisms, Clostridium and I think Acetobacter. So they're going to try to quantify the rates of transformation and then see if this could be important under certain conditions.
Okay, maybe a follow-on question for you, Alex, that's related to this. How long do you think extracellular DNA, I guess the question is with AMR genes, I guess it's with or without AMR genes, but how long do you think extracellular DNA with AMR genes would survive in the environment?
Right, so there's a bit of controversy about that because it's hard to study. So the DNA, when it's exposed, it will be degraded fairly quickly. Particularly if it's exposed to UV light. So it can degrade very quickly, but it can also be protected under certain conditions. So, for example, DNA can bind to soil particles and be less available to attack by enzymes and UV and other elements. But then if the DNA is protected, then maybe it's less easy for it to spread and transmit AMR genes as well. So the viability of DNA in the environment will vary depending on the conditions, vary depending on how exposed it is. And again, it's not very well known how long the extracellular DNA, even if it's present, it'll be breaking down. It'll be breaking down and becoming less intact over time. So we don't know how long it stays viable for transformation. But the PI of this project really believed that he's basically suggests that it's a bigger risk than currently, like the DNA is viable for longer than we expect, but obviously it will be viable for less time than DNA inside bacteria cells, of course, because it's being degraded. But again, like just to conclude, it depends on the conditions. Depends on how exposed it is to different conditions, into different.
Okay, all right. Thanks very much for that. So, Liam, a question for Liam, and I guess Dervla might want to chip in as well. I suppose a couple of linked questions. So, detection of antimicrobial resistance organisms in recreational waters, you discussed that. So firstly, are bathing waters not monitored, or are they monitored currently for the presence of such organisms?
So bathing waters are monitored for fecal indicator organisms, but they're not actually monitored for antimicrobial resistant organisms or other specific pathogenic organisms. So we will have a good idea of at a point of time when the water sample is taken, what is the risk that there is fecal contamination of that particular water body, but we don't really know what kind of organisms are going to be present there just by, I suppose, the numbers of E. coli or other fecal coliforms that we look for in the sample. And another thing to consider, I suppose, is that there's been recent evidence, and Dervla can elaborate on this, as to the methods that are currently used may not be great for they may not be a good indicator of what the kind of level of contamination of that water is over a period of time, because really only one sample is kind of taken over, I think it's once a month, isn't it, Dervla?
Yeah, so it depends on the particular local authority area or the particular schedule for sampling. But generally, bathing waters are just monitored for fecal contamination through the bathing water period, which is typically May to September. And the way in which the data is, or the way in which water is defined as being of excellent, good, poor, sufficient quality, is based on the total number of E. coli in a 100 ml sample. So 100 mils from an entire ocean isn't a huge volume. So through previous EPA-funded research we carried out a number of years ago, we showed that taking a larger volume is more accurate in determining what the risk is associated with that water. So in the ARREST project and also in other projects, we actually collect a 30-litre sample and we filter it. And again, if you're just looking at the total number of E. coli in a 100 ml sample without looking at the characteristics of that E. coli, it doesn't really tell you much about the risk to public health. So, you know, again, we found antimicrobial resistant E. coli in waters that were defined as of excellent quality. So it brings into question whether or not there's a need to revise the current EU bathing water monitoring criteria to include, you know, looking at things like antimicrobial resistance. And also, as I mentioned in my presentation, there is a significant need for a more systematic surveillance of the environment for antimicrobial resistance to feed into those One Health reports that would include not just animal and human data, but also environmental data, so that you can close that gap in the One Health paradigm that currently exists. And it is a focus of attention, and not just by us, but by several groups across the world.
And I suppose there's a linked question from somebody else asking, does the detection of AMR organisms in recreational waters, will that have an impact? You've sort of touched on this, but will it have an impact on future monitoring and how monitoring will be done for contamination?
So I suppose that's what we're hoping with the ARREST and the peer projects, is to generate evidence that will inform policies in this area. When we initially started the ARREST project and I was presenting it at various meetings to policymakers and regulators, the question that always arose was, what's the implications for public health? Can you define a safe level of antimicrobial resistance in recreational waters? So through linking the ARREST and the peer projects, we're hoping to be able to answer those questions essentially and feed into policy in this area.
Okay, thanks very much. Okay, looking through the questions. Let me see. There's a few more have come in. I'm just trying to keep up with them. There was one question come in. I'm not sure, is this for Dervla or how are or possibly Liam, how are CPE infections being treated in hospital?
Go ahead.
So it depends very much on
the obvious answer.
It depends on the type of CP organism. So there's various different types of, so the CP or the C part of CP stands for carbapenemase producing Enterobacteriaceae. The carbapenemase enzyme varies greatly. So as Owen mentioned in some of these targets he'll be looking at in the WorldCom project, he mentioned KPC, NDM, OXA48. The resistance conferred by those genes varies widely. So for example, OXA48 may actually not cause any resistance at all to antibiotics, whereas NDM, for example, typically knocks out pretty much all of the antibiotics that we have available. So currently, thankfully, in Ireland, the majority of infections are still treatable. They will retain susceptibility to a number of agents. even with the most resistant of the CPE, the NDM, as I mentioned, they can still retain susceptibility to 3 antibiotics, tigecycline, fosfomycin, and colistin. And they can and have been used successfully to treat infection. But unfortunately, there are reports emerging in the literature and at conferences of failures and resistances emerging to those 3 antibiotics as well. So that's why we need to control the spread of these organisms. And there's huge work ongoing in Ireland, as Liam mentioned. It was the CPE were declared a public health emergency in Ireland in October 2017. The National Healthcare Associated Infection and Antimicrobial Resistance Task Force were established, and there's huge controls in hospitals in terms of trying to minimize the spread of these organisms.
And again, another question on the same topic has come in from a couple of different people. Do you expect to find many swimmers colonized with these AMR organisms? And if so, does that mean they're at risk of infection? Now, if I can add to that, is there anything swimmers or recreation people enjoying recreation time in rivers or the lakes or the sea can reduce or minimize their risk of becoming infected? I can't imagine there is.
Well, I think it's very important to state first out that, you know, we're not trying to discourage people from enjoying swimming in the sea or swimming in rivers or wherever your local bathing water is. I mean, the health outcomes from actually swimming and enjoying nature far outweigh any risks that you might have of picking up an antimicrobial resistant bug. But if we show via the PEER project that there is a risk of people becoming colonized from using the water, then, you know, that can feed into policy and hopefully we can control better how these organisms are ending up in our bathing waters. As far as what kind of risk people might have or what we're expecting to see, so the study that was done by our collaborators in the UK found that about 5% of surfers were colonized with the drug-resistant organisms versus, I think it was less than 1% of people who didn't regularly surf. So that was a number of years ago, and that it was in the UK. So we're not sure what we're going to find in Ireland, but I guess that's why we're doing the study, to find out. If they are colonized with them, for the most part, there's no main real problem with it, in that there's no recommended therapy to get rid of a bug that's colonizing your gut, because even though these bugs are antibiotic resistant, most of them aren't really inherently pathogenic, and they will just live happily inside your gut, much like all of the other E. coli that are living in there. The only problem really that can occur is if they get out of your gut and they get into normally sterile sites such as your urinary tract or your bloodstream, and then they can cause an infection which can be difficult to treat. And I suppose the more people that have these bugs in their guts, the more likely they are to get spread around in their environments and possibly be picked up by other people who may be more vulnerable to infection.
Okay, thanks very much for that. So maybe a few final questions from people. I'm just looking at the clock here. So I'm going to link 2 separate questions together for Owen here. First of all, we've had a couple of people asking, how does the LEC-LAMP methodology work? So if you can give us a very simple summary of how the LEC-LAMP technology works. And can I add to that another question that came in? Would LEC-LAMP be suitable for use with, for example, COVID-19 diagnostics? I mean, I'm aware for family reasons of the Dublin Airport LAMP testing that's been set up, as well as PCR testing. So is there a potential role for LEC-LAMP in COVID testing?
Sure. So much like PCR, LAMP uses short DNA primer sequences to target and amplify your gene of interest, and it does this via DNA polymerization. So what we've done is taken the standard LAMP method, and there's 2 main modifications. We've added in a novel cleavage enzyme, and we've also added in a novel probe. As for the probe, we've essentially taken one of the LAMP primers and modified it to incorporate a cleavage site, which is flanked by a fluorophore label and a quencher. So when this binds to your target, you'll get cleavage, and then you'll get fluorescence and signal production, and that allows for your detection. Now, as for point mutation detection or differentiation, or single nucleotide polymorphism detection, what we can do is design the probe so that the cleavage site in the probe can target this mutation. And then that mutation close to the cleavage site prevents cleavage and prevents fluorescence production. And this allows for your differential detection. So as for applying LEC-LAMP to diagnostics in the current COVID situation, there's a lot of recent research that has identified that there are various point mutations associated with COVID that are associated with more severe infection. So we could use LEC-LAMP to identify these COVID strains that are associated with more severe infection. And this would be very useful in terms of epidemiological monitoring and maybe even intervention in that. And again, this could be used in more point of care settings, like you mentioned, in airport settings and that.
Okay, and maybe a question which could be, maybe there could be contributions from Alex, Dervla and Liam into this. I mean, in terms of thinking of antimicrobial resistant organisms and potentially DNA that could be transmitted or transferred to organisms or people in the environment. Are there measures that farmers or public health people can put in place, I suppose, to reduce the risk of the transmission of let's say recreational swimmers or people who are exposed potentially even through drinking water, I guess, in the environment, so that their risk of being exposed is reduced? I mean, it'd be a time of year of spreading slurry or other measures.
I think it comes back to understanding better what waste treatments are going to work actually to reduce down the load of resistant bacteria and also genes and mobile genetic elements, etc., in the waste. So through the ARREST project to date, through nice work carried out by Sean Wang and Jim at NUI Galway, we've shown that anaerobic digestion is effective at removing CPE after about 7 days, depending on the amount of total solids present in that waste. Again, integrated constructed wetlands based on work we did with colleagues in the Department of Agriculture, Food and Marine at Back Weston, showed that they seem to remove, we found antimicrobial resistant organisms in the influent, but nothing essentially in the effluents from a year-long study. So I think we need to do further work looking at conventional wastewater treatment as well, and look at innovations in waste treatment to see how they might be applicable. I don't know if the others want to comment further on that.
Liam or Alex want to add anything?
My comment would be, if we can all look after how we're disposing of our antibiotics. So obviously there's wastewater treatment and how they're disposed of then, but even just for yourself, if you are taking an antibiotic course and you have some antibiotics that are left over, bring them back to the pharmacy. Don't flush them down the toilet, because they're just going to end up in waste there, and definitely don't throw them out the window. So I think, if there isn't antibiotics in the environment, you're really reducing the selective pressure, like Alex said, for these bugs to develop the resistance.
Okay.
I suppose the only thing I would add is that, like Deborah was saying, we need to understand the things a bit more about transmission of AMR genes in the environment. So in terms of neutralizing DNA as a possible source for AMR genes. If the AMR genes are present in extracellular DNA in the environment, we don't know yet how risky that is. I think that's what we're trying to find out. If it is a risk, then we're going to have to look at ways to minimize, because of course DNA will be degrading over time. There may be treatments that you can do to reduce, to break it down further or faster. But at the moment, we need to assess the risk. Is it a risk, and when is it a risk? So I think that there's more research that needs to be done there.
Okay, thanks for that. And actually, that's very good timing now, because we've just come up to 4 o'clock. So we've come to the end of our 90-minute webinar. So can I just thank first of all the speakers for their talks. Can I thank you, the participants who attended the webinar and who contributed, those who contributed questions into it. Hopefully you found it interesting, and hopefully you found it informative. I mean, I think it's certainly given us all, including certainly myself, insights into the complex world of antimicrobial resistance and I think the value of the One Health approach. So can I finish by just thanking a few people for their fantastic work, the organizing committee over the last number of weeks, firstly to Liam Burke, Louise O'Connor, Georgios, and the Ryan Institute communication team, in particular Isabel McLaughlin and Nora O'Halloran. So, as I mentioned at the start, this webinar is part of our Centre for One Health Spotlight series. So please keep up to date. We'll be communicating with people through the communications team, but please keep up to date about future events in this series and other Centre for One Health activities by logging into our website. Please also follow us on Twitter. You can find out about us via Eventbrite, or drop us an email at onehealth@nuigalway.ie. We have recorded the webinar on YouTube, and it will be available very shortly on our YouTube channel. So thanks again for your attendance, and look forward to hopefully seeing you all at our next event. Thanks very much, everybody.